Nestled in the campus of Auburn University in eastern Alabama is the Auburn Museum of Natural History.
The Museum of Natural History is primarily a research and education museum. You might have noticed when you came in, we don't have a giant, whale hanging from the ceiling. There's no display space. Hopefully in the future we'll get that.
David Laurencio is one of the resident herpetologists at the Auburn Museum of Natural History. Herpetology is the study of reptiles and amphibians, which includes animals like
Salamanders can lose their tails
salamanders,
back, so this one lost his tail, but not right now.
turtles,
Oh, there you are.
lizards, and frogs. Though David studies a variety of reptiles and amphibians, his main research interest is in frogs.
Well, there are some characters we can use to distinguish the main groups of frogs here in Alabama. We're very lucky to have lots of species here. We have over 30 species of frogs in Alabama. The toads are identified by two little bumps on their necks, and those are actually glands that produce toxins that the toads actually use to help for defense. But all toads will have those two little bumps on the back of their neck. So most of our toads are brownish with kind of warty skin. It's very dry, but they'll all have those two little glands on the back of their neck. Our tree frogs, as their name implies, do climb trees a lot. So they have characters and adaptations that help them do that. The most noticeable are their toe pads, and they're basically little sticky discs that help them to climb up on trees and hang on. And this is why you might see a tree frog hanging on your window at night, because they can use these discs even to hold on things like glass. And this goes along with their lifestyle of living up in the trees.
But of course, it also allows.
And then the true frogs, the ranids, which are frogs that we might recognize like our bullfrog, our banjo frog, our leopard frog. This is a highly aquatic group. So they're going to have adaptations or characters that help them live in and around water. So they've got really long legs, meaty legs too. If you've ever had frog legs, they tend to come from this group. They'll have extensive toe webbing, and that webbing between their toes helps them to push against the water and swim. And then their body is kind of streamlined to be able to glide through the water easily. But those are kind of our main groups. So frogs are kind of neat. They're very neat, I think. But frogs are not unique, but they share the character with birds that their frog calls are unique to species. So every species of frog has a frog call that is unique to itself. And that's how the females can recognize males of their own species. It's the males that call, and generally the calls that we hear at night in ponds and ditches or streams are males calling to attract females. And that's how the females can recognize males of their own species. And the neat thing is, each frog has its own call. Our bird voice tree frog, for instance, has a very high-pitched frog, so you can use the pitch. The type of call, from a bleat to a trill to a quank. In some cases, the call is very unique. Like the leopard frog has a chuck and a chuckle that's put together, which is kind of neat. Some of our toads just have really long trills. The bullfrogs have a very low call. They can be distinguished really by listening to them and finding uniqueness in their call. Just professors and researchers in general will come and use our collections, looking at everything from diet to morphology to coloration, reproduction.
So, where's the right here.
Andrew Collins, a researcher from Jacksonville State University, has come to the museum to collect measurements of different species of frogs for his graduate project.
Hi, I'm Andrew Collins. I'm a grad student here well, I graduated here from Auburn, and now I am a grad student at JSU. I'm going to be doing my research on Hyla gradiosa, which is the Barker tree frog, and Hyla cinerea, which is the green tree frog. And I'll be looking at the hybrids between the two up at Frog Pond up in northeast Alabama.
And then we'll also have a collection of genetic material that's used for molecular studies, looking at things like phylogeography or the diversity within or among groups of amphibians and reptiles. And this means that we no longer have to take a piece of tissue from an animal itself, say the frog or salamander. We can now actually go collect water from a stream and then test that water for the presence of the DNA of a given species.
Environmental DNA, also known as eDNA, which uses existing DNA of organisms and compares them to the DNA found in bodies of water, is a recently developed biological tool that has helped to locate and study rare or hard-to-find species.
Using eDNA allows us to check several ponds or streams for these with relative ease compared to actually finding the larval forms or the adults of species that are so rare. It's kind of opened up a new tool for conservation here, not just in Alabama, but everywhere.